Hello everyone, this is Ketish here. In this video, I'm going to go through the AQA 2023 Biology Paper 1 Higher Tier, question number 3, which is testing on the topic of infection and response. Salmonella bacteria can cause outbreaks of food poisoning in humans. To prevent food poisoning in humans, farmers vaccinate their animals against Salmonella bacteria. How do Salmonella bacteria in food cause the symptoms of vomiting and diarrhea? Now, the reasons for these symptoms is because the bacteria produces or releases toxins, which are waste products released by the bacteria as they reproduce and carry out their normal functions. And the toxins are usually released to discourage other bacteria from growing. So it's a way of survival for the bacteria, but unfortunately for us, it causes vomiting and diarrhea. During a food poisoning outbreak, scientists identified the farm where the food came from. The farmer had not vaccinated the farm against animals against Salmonella bacteria. The food poisoning outbreak could have been prevented if the farm animals had been vaccinated. Explain how the immune systems of animals respond to a vaccination. It's the same way that animals would respond. The immune response in farm animals prevents an outbreak of food poisoning in humans. So it's in two parts. How does vaccination affect the immune system of animals? Now, remember, a vaccine contains dead pathogens or parts of the broken down piece of the pathogens or the antigens. So when vaccinated, the animal's white blood cells, let's say in sheep, cattle, or even chicken, the white blood cells, which is basically the lymphocytes, produces the antibodies against the antigens. The antibodies are very specific to the antigen, which is basically complementary in shape, the opposite in shape, and specific antibodies bind to the Salmonella bacteria or to the antigens. Now, the secondary response, here's the answer in detail. So it's a bit of a long answer. You can shorten it. You're not expected to write all of the points. Right? Animals' white blood cells, the lymphocytes, produce antibodies against the Salmonella vaccine or the antigens. The antibodies are very specific or complementary in shape. Specific antibodies bind to the Salmonella bacteria. The secondary response in animals, if infected, specific antibodies are produced quickly or in large numbers. You could mention by memory cells. So the white blood cells or the antibodies would kill the live Salmonella. So fewer or no bacteria pathogens would survive in animals which we are using to produce meat, milk, or eggs. Prevention of food poisoning in humans. Now remember, the answer, second part to the answer is the immune system in farm animals prevents an outbreak of food poisoning in humans. If the food is not poisoned, then we will not pass it on to each other. So fewer or no bacteria or Salmonella is eaten in foods like milk and eggs. So number of bacteria never reaches a high level for infection to develop in humans. So fewer toxins produced in humans. Remember, for level 2, students must link immune response in animals to prevention of an outbreak in humans. That means to gain 4 marks, you need to link the immune response to the outbreak in humans. Most cases of food poisoning do not need to be treated with antibiotics. However, some patients may need to take antibiotics to recover. Scientists investigated the effectiveness of 5 different antibiotics on the Salmonella bacteria in the outbreak. Antibiotics A, B, C, D, and E were used in the investigation. Figure 4 shows the results. So you've got a petri dish with agar gel, and in that we've got the bacteria, Salmonella bacteria, growing, and paper disc containing the antibiotics. So we would have tiny paper discs, like using a hole puncher, and we would soak them in antibiotics and then place it inside the petri dish. Paper disc with no antibiotic is actually in the middle. Describe 2 aseptic techniques the scientists should have used in the investigation. Now, while you're trying to set this up, you need to make sure the first thing that you need to do is actually set up the petri dish with the agar plate. For that, you need to use aseptic techniques. The bacteria can be cultured where you can actually buy them in liquid form, so there's not a lot to be used there. But when you're setting it up, you have to follow aseptic techniques so that you don't get any other pathogens or fungus from the air landing on it and growing. So here's any two of these will be fine. Disinfect hands and the work surface before you start. Sterilize petri dish or culture medium before we use. So sterilize means basically you place it in boiling water in a pressure cooker. Pass inoculating loop or forceps through a flame. So usually if we are, let's say if we place the paper disc on the agar plate and then placing a couple of drops of the bacteria solution, or you have to use small tweezers to place it, so you need to pass that over a blue flame. Work near a flame or work inside a fume cupboard. Right. Tilt the lid of the petri dish when opening this, placing this on agar to minimize contact with air. You don't completely open it, you just open a little bit and then place the disc inside. Secure the lid of petri dish with adhesive tape. You don't completely seal it off, you just seal it off so that the lid doesn't actually fall off. The scientists incubated the bacteria at 37 degrees Celsius. Students in school laboratories incubated bacteria at 25 degrees. Explain why scientists use 37 degrees Celsius, but students must use 25 degrees to incubate bacteria. Now, 37 degrees is the human body temperature. So you don't want to encourage any bacterial growth that is very similar to the human body temperature, especially in the school lab. If it is in a proper scientific lab, then you could actually grow bacteria. They will have license to grow bacteria at that temperature because they would have the adequate safety measures, which we usually don't have in schools. So 37 degrees Celsius is human body temperature. The Salmonella bacteria grows best at this temperature. 25 degrees Celsius reduces or prevents the growth of bacteria that are harmful to humans or to the students. What is the purpose of the disc, paper disc with no antiseptic in figure 4 that is used as a control? The scientists concluded that either antibiotic A or antibiotic B should be prescribed to patients with food poisoning. Why should antibiotic A or B should be prescribed? Now, if you look back in the diagram, you can see the clear area is where the bacteria is actually not growing, because they have labeled Salmonella bacteria is growing in the gray area. So where you have the clear area, there is no bacteria growing because of the antiseptic slowly soaking into the agar gel. So you've got A and B with the biggest circles of clear space. So A and B could be used because they killed the most bacteria. The scientists wanted to be more certain about which antibiotic should be prescribed. Describe how the results in figure 4 could be used to obtain a quantitative comparison of antibiotics A and B. Now, quantitative means you're trying to quantify something and to measure how much. So basically you want to measure in terms of numbers. Now, the best way is actually measure the diameter of the white circle in the petri dish. So measure the diameter or the radius of each clear area to calculate the area for, or measure the area for each clear area. 1 year later, there was another outbreak at the farm involving Salmonella bacteria. Antibiotic B did not have an effect. Suggest why antibiotic B no longer had an effect. Main reason antibiotics do not are not effective against bacteria is because the bacteria would have become resistant to that specific antibiotic. Antibiotics treat food poisoning because they kill salmonella bacteria inside the human body. Some antibiotics work because they damage the bacterial cell wall. The bacteria die because the cells burst. Explain why the cells burst. Now, if the cells are bursting, that's mainly because the cell is actually getting bigger and bigger. So, if the antibiotic damages the bacterial cell wall, then water naturally would go into the bacteria by osmosis. When it does, it would increase the size of the bacterial cell. The cell wall usually should protect it from bursting. Without the cell wall, the cell membrane will simply rupture because of the increased pressure from the water. That brings us to the end of question number 3, and I will see you in question number 4.